Welcome to the Special Senses lecture. This lecture will cover mostly the anatomy of the special senses and touch on some of the physiology as well. So the five main special senses are taste, smell, sight, hearing, and balance. Under balance, you have the sense of touch as well, but touch is not considered a special sense, so we consider it a general sense in itself. We'll start with the chemical sensations first. You have taste and smell, which are both considered chemical senses because both of them require putting chemicals into your saliva or the mucus in your nasal cavity to create some sort of reaction. Now, you have special receptors that are able to sense taste and smell chemicals, and these are called chemoreceptors. Chemoreceptors are simply a class of receptors that bind to chemicals to send a signal to your brain. Now, for taste, the food needs to dissolve in the saliva in order for it to bind to chemoreceptors. And then in smell, the sensation of smell or the chemical associated with that smell needs to dissolve in the nasal mucosa in order to bind to chemoreceptors. So the first sense we're really going to get into is taste. Be sure you're aware of the illustrations that I'm pointing out so that you can use those to study for the lab exam. So for taste, the anatomical term for tasting is gustation, and gustation is done using taste buds. Now, if you've ever looked at your tongue in the mirror, you see those little dots on them, they're little raised bumps. These bumps, these are not individual taste buds. These are called papilla. This is a papilla, that's a papilla, this whole row of big round ones is papilla. The ones in the back, these are not papilla, these are tonsils, so we're going to leave these alone for right now. So the two types of papilla we're going to cover are fungiform papilla and circumvallate papilla. Both types of papilla are going to be found on your tongue, and they both have taste buds surrounding them. The ones that are found on the body of your tongue are the fungiform papilla. You see them labeled here. And the ones that are on the back of the tongue, the base of the tongue, kind of going in a line, these are circumvallate papilla. The other two types of papilla, they are also present on the body of the tongue, but we're not going to talk about these right now. So the fungiform papilla and circumvallate papilla, they both have taste buds surrounding their perimeters, but as you see, the circumvallate papilla are larger, so they're going to have substantially more taste buds because they have a larger surface area surrounding them. You have way more taste buds that are in the circumvallate papilla than are on the fungiform papilla. This means that you're going to get a much stronger taste sensation back here at the base of your tongue. Now, taste buds, they are made up of two major types of cells inside each bud. You will see what each bud looks like in just a moment. The two cells are gustatory cells and basal cells. The gustatory cells, these are the actual cells that have the chemoreceptors on them to actually bind to the taste chemicals. The basal cells, these are your helper cells that can help repair damaged gustatory cells, or they can just simply act as supporting cells. These cells, the gustatory cells, have very long microvilli. And if you remember what microvilli look like, they look like this. And these microvilli, let's say this is a gustatory cell, they have little tiny receptors on them that we call chemoreceptors. And these chemoreceptors are what bind to the taste chemicals. Now, all taste buds are replaced every 7 to 10 days, and taste buds are responsible for tasting things that are sour, sweet, salty, bitter, and umami. Now, here's the anatomical illustrations of it. This is your entire tongue. You've got a dispersion of the papilla on the body of the tongue, and then you've got a single line of the circumvallate papilla on the base of the tongue, and you've got your lingual tonsils back here. What we're going to do to learn the anatomy of a papilla and a single taste bud is by taking one large circumvallate papilla, this right here, that's your circumvallate papilla. You see that around the perimeter of it, you have these deep pits. These deep pits is where the food that you eat dissolves into the saliva and creeps down into these deep pits. Because if you see on the edges these purple buds, those are all taste buds. So the whole thing is a papilla. Each individual purple head is a taste bud. You notice the taste buds are connected by these yellow pathways, and those are nerves to go to your brain. Let's take a zoomed in look at one taste bud here. So there's the nerve in the back of it, and here's that purple head. Remember that taste buds are made out of two types of cells, the gustatory epithelium and the basal epithelium. The basal epithelium are usually at the base of the taste bud, and the gustatory epithelium usually go out towards this opening here, and this opening opens up towards that pit that we saw. So this pit here, that's where your taste buds open up to. In these openings, and it's not very clear on this illustration, so I'll draw it in, you have these little hairs, those microvilli that stick out, and these are where you have those chemoreceptors so that you can bind to all the chemicals that are coming and sinking down into this area. So when this taste chemical binds to the chemoreceptor, it sends a signal to the back of the gustatory cell and eventually down the nerve to your brain. Now, the pathway that we're going to take is going to be this list here. Make sure you know this list, please. This is the pathway that we take from this nerve all the way to the brain. These guys are all cranial nerves. Cranial nerves are always denoted using Roman numerals. So, cranial nerve and cranial nerve. This is cranial nerve number 7. This is called the facial nerve. Cranial nerve number 9 is called the glossopharyngeal nerve. Both of these nerves bind in your tongue to send signals that'll eventually go to your brain. So we first take the facial and glossopharyngeal nerve, and we're going to send that signal to the medulla of the brain. That's in the brainstem. From the medulla, we travel to the thalamus. From the thalamus, we go to the cerebral cortex. Just to give you kind of an idea of what this looks like, let's say the brain looks like this. And then we've got the cerebellum back here. We've got that. We've got the brainstem. So let's say this is your tongue. You have cranial nerve 7 and 9 traveling to the medulla. From the medulla, we're going to go up to a round structure called the thalamus. And from the thalamus, we send signals out toward the cerebral cortex. And the cerebral cortex is just this area here, the cortex of the cerebrum. This is where you do conscious integration of information. So you can consciously say, hey, I just tasted something bitter, I don't like it. Or hey, I just tasted something that's way too sweet, it's too sweet for me. So make sure you know this pathway. The next sense is going to be smell. We call this olfaction. Olfaction is done in the nasal cavity, not in your nostrils. You're going to go all the way up your nostrils, far back after you can even reach picking your nose, and enter the nasal cavity. And inside that nasal cavity, which would be somewhere between your eyes, between your nose, if you took a drill and drilled right back, there'd be an opening in there. That nasal cavity is covered with mucus. The top, and listen here, only the top of that nasal cavity has olfactory epithelium on it. Olfactory epithelium is the only place you're going to find the chemoreceptors for olfaction. We are going to see a picture of this on the next slide, so just bear with me. Olfactory epithelium is made out of 3 different cell types. Olfactory sensory neurons, of course you've got your supporting cells, and olfactory stem cells. These 3 are going to be found on this top layer. So let's look at this illustration here. You smell something, goes in through the nostril. This is about as far as you can pick your nose right here. If you keep going far to the back, you're going to have this large opening. This whole opening is covered with mucus, but the top of it, where you have the ethmoid bone, do you remember that ethmoid bone? is that cribriform plate. On this bone, on the bottom of it, you're gonna have the olfactory epithelium. This is where all of those little microvilli and cilia stick out to bind to the chemicals of smell. Eventually, they bind to the chemical of smell, send that signal up and to your brain. Remember that you have to have mucus to dissolve the chemical first, just like how in your mouth you had to have saliva to dissolve the chemical first. So this is a very, very good illustration for the lab practical. It's very detailed. And we're just going to start with, again, going over the pathway. You smell something. It goes into the nasal cavity. It goes up to the top of it, binds to the chemoreceptors of olfaction. This is where we're at right now. So here's the top of the nasal cavity. You see the mucus, and you see the little cilia sticking out. It's on this cilia that you're going to have tiny little chemoreceptors. So the scent dissolves in the mucus, binds to the chemoreceptor, travels up the cell, which is this yellow one here. This yellow cell is an olfactory sensory neuron. I'm going to circle that for you. Olfactory sensory neuron, that was one of the cells from the last slide. The ones next to it, the pink ones, are the supporting cells, and the blue ones are the stem cells. The main one we need to focus on here is the sensory neuron. So the chemical binds to the cilia, travels up the sensory neuron, and it keeps going up until it passes through this bone. You can tell this is a bone here. And this is the cribriform plate of the ethmoid bone. If you remember in lab for bones, the cribriform plate was very porous. It wasn't very smooth or solid or anything. And that's because you need pores to let these neurons pass through. Eventually, these olfactory sensory neurons are going to come and converge at these orange cells called mitral cells. This area right here that I'm circling, that conversion point, these are all called glomeruli. Okay, I know you've seen this word before in the kidneys PowerPoint, but you're going to see it again. This is the glomeruli of olfaction. So the signal goes up the sensory neuron, sends a signal through the glomeruli into the mitral cell. The mitral cell converges all of its axons together, and that makes the olfactory tract. This olfactory tract will eventually go to the cerebral cortex and what we call the limbic system. You saw the cerebral cortex, which is the outside of the brain, and it's going to be kind of in this region here, going all the way around. And the limbic system is going to be a system of neurons that also have to do with memory. This makes you wonder, is this why scents are so heavily tied to memories? And the answer is yes. Because what you smell makes its way through the limbic system, a system that has to do with memory as well, you tie in certain memories with what you smell during that time. Like for me, I'll never forget the smell of those paper towels in public bathrooms. You know, the kind of brownish paper towels. A wet brown paper towel to me smells like kindergarten. I will never forget like being in the bathroom in kindergarten. As young as I was, this sounds crazy, but I will never forget standing there, smelling my wet paper towel, thinking, wow, I'll never forget this smell. And I never did. There are certain smells that we all smell that take us back to a certain time or a certain memory. And that's because this limbic system, responsible for making memories, is also responsible for integrating olfaction. So kind of cool. Now moving on to the main big one we're going to spend a lot of our time on. Now I'm kind of bummed out because we would have dissected an eyeball in lab, which is so cool because it's a cow eyeball and you can see almost all the features on it. So from your list, from the lab list, do not worry about needing to know the eyeball dissection. If I can find a video on a cow eyeball dissection, I will post it and let you guys all know. But for now, don't worry about the cow eyeball dissection or labeling it. We're just going to focus on the illustrations that I have for you here. So Why do I love vision so much and the sense of sight and the eye in general? Well, because of these two big facts. 70% of all of your sensory receptors in your entire existence is in your eye. 40% of your entire cerebral cortex is going to be involved in visual processing. So you're spending a lot of time, energy, you've invested a lot into just vision. So let's start from the outside and work our way in. Kind of outside the eyeball, on the lateral part of your ocular region, you have this gland called the lacrimal gland. The lacrimal gland is responsible for lubricating and making tears for your eyes. It secretes those tears from the lateral side. Those tears work their way to this middle region called a punctum, and they'll get collected through this lacrimal sac, down the lacrimal duct, through the nasal lacrimal duct, and out of the nostril. If you ever notice when you're crying, you end up feeling like you have a runny nose. It's not because you have a runny nose. It's because you're hyperproducing tears that get collected and drained out of the nostril. Make sure you know this illustration. This is the exact one that I will use for your practical. This illustration as well is phenomenal. Usually I test on a model on this one for the lab exam, but we're going to use this illustration here. This illustration will show you the nasal bone. This is a right side eye looking from a lateral view. And the eyeball here is connected by several muscles, specifically 6. And you have to know those 6 muscle names. So I'm going to circle them for you. So these 6 muscle names might sound scary, but they're actually very, very easy to memorize because you basically know all the directional terms for them already. We're going to first talk about the rectus muscles. There are 4 rectus muscles, and the rectus muscles bind to the eye with a tendon, and they pull straight back. So we've got the superior rectus muscle, and if you've got a superior one, what's the one on the bottom going to be called? Inferior rectus muscle. So we've got a superior and an inferior rectus muscle. The one on the outside is called the lateral rectus muscle. And if there's a lateral muscle, there's also going to be what? A medial muscle. So lateral rectus muscle. And the medial rectus is right here. Let's see if I can write this down. It's very difficult to use this laptop. Medial rectus. There we go, not too bad. Then you've got the oblique muscles. So we covered 4 rectus muscles, and then we got 2 oblique muscles. The term oblique means to kind of go in a circular orientation like that. So you've got the superior oblique muscle, which binds on this way and kind of makes a U-turn. And you've got the inferior oblique muscle, which kind of wraps around this way. So superior and inferior oblique muscles. Make sure you know the names of these 6 muscles that are found around the eyeball. And it is the job of these muscles to turn the eyeball in different directions. This is the same picture, just an anterior view. I can use this one as well. You see the rectus muscle superior, inferior, medial, and lateral. For you to know medial and lateral on a picture like this, for you guys, I would have to tell you which side the nose was on. And then you've got the superior oblique and the inferior oblique. Now, the eyeball itself has a lot of anatomy. The anatomy of the eyeball, we're going to start from the outside and work our way in. This is something that we would have done using the eyeball dissection, so we're just going to use the illustrations for this one. Now, the eyeball, on the outermost region of just the eyeball, if you were to extract an eyeball from a human, a cow, any kind of animal, it would be covered with a very, very rigid fibrous material, which we call a sclera. And let me get my laser pointer out again. The sclera doesn't cover the entire eyeball, of course, because the front of your eyeball has the opening called the cornea. It is a clear region. So the majority of the eyeball is covered with a sclera. This is usually white, And this is where all those 6 muscles that we talked about anchor to. The front of the eyeball, where you have the color showing, where light enters from, that's covered with a specialized tissue called a cornea. The cornea allows for light to enter into the eyeball. And so if I can just show you here, you've got the sclera, which is this white fibrous tissue. It's pretty tough. If you guys were cutting into the cow eyeball, you'd realize that it's pretty hard to cut into. You'd have to go back and forth a few times to actually get into it. And then in the front of it, this, what's represented in like a light blue, it's supposed to represent it being transparent. This is the cornea. So it starts here and comes all the way out to right there. This is probably my favorite illustration of the eyeball because every single thing on here is on your list except for central artery and I don't think suspensory ligaments is on your list either, but double check that one. So let's go through the anatomy of this. In the coming slides, you have functions of all these guys, but let's just go through it together just using an illustration. So you've got the sclera in white. You've got the cornea in light blue. If you notice underneath the sclera, you have 2 more layers. You've got the choroid layer, and this is vascular. You can see all the blood vessels going through it. And then the yellow layer is the retina. In real life, the retina is actually white. It's not yellow, but for teaching purposes, we put it in yellow here. So this layering, again, sclera, choroid, and retina. You see that it's the same consistently throughout the posterior of the eyeball. And then inside, it's covered with this jelly. It's full of this kind of like jello-like material. And this is called vitreous humor. Vitreous humor is the jello-like filling found in the posterior of the eyeball. And with that, let me just introduce to you the fluid that's found here. The fluid that's found right behind the cornea, but in front of the lens, this is called the aqueous humor. So easy way to remember it is aqueous starts with the letter A for anterior. This is the anterior compartment. This is the posterior compartment. So aqueous humor is in the anterior compartment. Vitreous humor is in the posterior compartment. So what's in between those 2 compartments? The lens. This is the lens of your eyeball. This lens doesn't just float here. It's tied with these suspensory ligaments. And it's tied with the suspensory ligaments to these little finger-like projections we call the ciliary body. And this ciliary body actually wraps all the way around the eye. And it wraps all around the lens as well. But because this is a mid-sagittal view of the eyeball, it just looks like it's holding from the top and the bottom. So again, in reality, these suspensory ligaments are actually all around the lens, and these ligaments allow us to contract or relax the lens because by doing that, you can control where the light actually gets into the eyeball too. This is something you probably won't learn unless you take physics. The opening that lets light come in is called the pupil. The tissue around the pupil, which is really the color of your eye, is called the iris. So people with blue eyes have a blue iris. People with brown eyes have a brown iris. And the opening in the middle of it is called the pupil. So far we've done everything in the front of the eyeball. Let's just go to the back here. In the back of each eyeball, you have a single nerve leaving. This is called the optic nerve. If you notice, the optic nerve contains the vasculature in the middle. It contains an extension of the retina, and it has the sclera covering it as well. This is where we collect all of the signals from the retina and we send it to the back, and this optic nerve eventually goes to the brain. The last 2 things on here are the macula lutea and the fovea centralis. The macula lutea is this dark, kind of brownish region. the macula lutea does pretty high acuity vision. Like if you're really, really focusing on something, you're going to reflect most of the light onto the macula lutea because that's where you have very accurate vision. In the middle of the macula lutea, take it a step further, you have a region called the fovea centralis. It's very, very small. But the fovea centralis does the highest acuity vision for you. We'll talk more about that at the end of this PowerPoint. So this is just telling you what each region does. If you remember, the posterior compartment was full of vitreous humor. And the function of vitreous humor is to help transmit the light. So with this jelly material helps to transmit light faster and more clearly to hit the back of the eyeball, which is the retina. This is where you actually are able to send signals of light. It supports the posterior surface of the lens, so it helps push back on this lens so the lens doesn't fall or flop down. And it also maintains the shape of this eyeball. Sometimes people have too much vitreous humor, it puts too much pressure in the eyeball, or sometimes people have too little vitreous humor or it's leaking out, and so the retina of the eyeball starts to collapse in just very slightly and it can cause blurry vision. So having the right amount of vitreous humor to help keep everything pushed back in its place, to keep the lens pushed in its place as well, is very important. The aqueous humor, that's in the anterior compartment, this is constantly renewed and it comes from your blood. So from the blood, we're able to extract things like proteins, sugars, and water. And we put those in the aqueous humor and it supplies nutrients to the lens and the cornea. Remember the iris, the colored part of your eye? The iris is made out of smooth muscle. The iris is able to contract and relax to either make the pupil dilated or constricted. To do this, the muscles that we use are called the sphincter pupillae muscle or the dilator pupillae muscle. We're going to see this picture in the coming slide. The reason that the pupil dilates and constricts is because you want to control how much light is actually getting into your eye. If you ever shine a flashlight into someone's eyeball, you will notice that their pupil constricts because the eyeball says, whoa, there's way too much light in my environment. Let me constrict my pupil a little bit to restrict how much light is getting in, because if you don't do that, too much light can damage the retina. So your eyeball has this way of saying, hey, it's too bright, constrict the pupil. Or hey, it's really dark outside. Let's dilate our pupil so we can get as much light in as possible. Then you got the choroid. I'm going to go back and show you the choroid really quick. This dark red layer. This dark red layer, the only thing I've told you about it so far is that it's vascular. What I didn't tell you is that the choroid is there to absorb as much light to prevent scattering of it. You don't want the light to enter into your eyeball and bounce around in there. You just want the light to enter in, go to the back, and absorb wherever it hits. There are animals like cats and cows whose choroids are shiny, and that allows light to come in and bounce around and create a stronger image, and it's usually doing that in the dark. So choroid color makes a difference. For humans, ours is just brown. Because it's brown and not opaque or shiny, we're not able to bounce around light, so we don't have very good night vision. But animals like cows, foxes, and cats, they do have opaque choroids. And you can tell this if you look into the eyeball of a cat when it's dark, they look kind of yellow and shiny. But if you look into a human's eyeball when it's dark, it sure as heck better not look shiny. So this is the slide that shows you the pupil dilation and constriction. So again, this colored region, this is called the iris. In the middle, the opening is called the pupil. So if we look at it on an eyeball, we've got a round muscle, and then we've got a muscle that kind of looks like a starburst. The round muscle is called the sphincter pupilla. And the job of this guy is to contract and decrease pupil size. The one on the outside This is called dilator pupilla. This is to increase the size of the pupil. You can constrict to restrict how much light gets in, or you can dilate to allow much more light in, and it usually does this in the dark. You do this as well when you're in your sympathetic nervous response. This response is your panicking response. Now think for a moment. Why would you want to dilate your pupils when you're panicking? What can you do better when you have dilated pupils? Think when you're panicking, let's say someone's chasing after you. Someone's chasing after you. You're looking straight ahead to where you're running. But what do you want to have just to see if they're catching up? The answer is peripheral vision. The bigger your pupil is, the more you can see to your vicinity and to your periphery. So your body knows, okay, hey, we're panicking. It might be better to have better peripheral vision to see if we're in danger still. Then you've got the retina. This is where you're going to find all of those special cells. The retina is made out of 2 layers in and of itself. On the picture that you saw, it just looked like a yellow tissue. But now I'm telling you that that yellow tissue is made out of 2 layers. One is called the pigment layer, which is made out of melanocytes. The other one is called the neural layer, and this is where you find all of your specialized cells for vision. The specialized cells we're going to talk about are photoreceptor cells, bipolar cells, and ganglion cells. The 2 types of photoreceptor cells that you have are going to be called rods and cones. Some of you may know what this is, but we're going to talk about their functions right now. So this picture is a wonderful picture, and I think on the next slide it's labeled. So we're just going to go ahead and use this illustration, and I'm just going to walk you through what everything is. So this is the front of your eye. The eyeball is kind of counterintuitive because you would think that, hey, when light comes in, it would stimulate this one, then that one, then that one, and then send the signal back. But that's not really what it does. In reality, in the front of your eyeball, light comes in, it passes in between all of these cells until it hits the melanocyte layer. This is that melanocyte layer. The signal is then transmitted back forward to this cell, from this cell to this cell, from this cell to this cell, and then that signal is sent out of the optic nerve to the back of the eye, eventually to the brain. So with this, I'm going to teach you the names of the cells, and we'll point them out with colors. In purple, you have your rods. In yellow, you have your cones. Rods and cones are there to perceive light. Cones work in the daytime, so we're going to draw a little sunshine here. And rods work in the nighttime. So rods are best for night vision, and cones are best for daytime vision. These guys also are best for color, and the rods are best for black. That cursive is easier because you don't have to lift the pen. Black and white. There. So rods and cones detect either color vision or black and white, daytime or nighttime. In either case, they will sense the wavelength of light that's hitting them, and they will send that signal to the next cell. This next group of cells are called bipolar cells. Those are the red ones here. The bipolar cells then send the signal to the next set of cells, which are ganglion cells. And ganglion cells will converge all of the signals together and send it to the brain. Now, in between the rods and cones and bipolar cells, you have this green cell here. If you notice, the legs of the green cell are converging the signals from the rods and the cones. Those are called horizontal cells. The light green cells Notice their legs are converging the signals between the ganglion cells. These are called amacrine cells. So rods and cones, horizontal cells, bipolar cells, amacrine cells, and ganglion cells. They go in that order to generate a signal, converge the signal, and then send the signal of vision to the back of the brain. Make sure you know the order that the cells are sending signals to, and remember that the light passes through the whole thing and then starts sending the signal forward. Now, in the back of the eyeball, you see that there is a region here that doesn't have any kind of cells. You see that you got cells, no cells, and cells. That region, because it doesn't have any cells, is unable to detect any light. Because it can't detect any light, we call this the blind spot. The blind spot, everybody has one, and it's there because this is where all of the converging signals actually need an opening to leave the back of the eye through the optic nerve. And that optic nerve, there's one from each eyeball, and they will eventually go to the brain to send the signal to the brain. So here's the definitions of rods and cones. Your rods, again, are in purple, and rods are more sensitive to light, so they allow vision in darkness. Cones, like daytime vision and color vision, so these guys are pretty high acuity vision cells, and they are both pretty evenly scattered through the back of the retina. Now, I promised we'd go back to this, and we are now. So this is an actual image of an eyeball, and here you can see the blind spot. And in this blind spot, you have the optic nerve leading out of the back of it. You see it of all the vasculature coming in and out of that spot as well. Right to the left of it, do you notice that this is darker? This darker region is called the macula lutea, and if you remember, the middle of it is called the fovea centralis. The macula lutea does high acuity vision, and why is that? Well, because it's mostly cones. It is not an even distribution of rods and cones here. It is mostly cones. And in the middle of it, the fovea centralis has only cones. So when you're doing high acuity color vision in the daytime, and a great example of this is the kind of vision you were using with histology slides. If you remember looking through the microscope, that microscope made your eyes pretty tired, and that's because your eyes kept trying to really focus the image onto this region of your eyeball, because this is where you do the most detailed, highest acuity color vision. And that's the end of the special senses lecture. It is just the tip of the iceberg of special senses, so when you guys get into physiology, you're going to go into everything that we did today again. And you're going to go into a lot more detail of how those cells function and what they do.